All right. Well, our first exam is coming up. So exam number one is going to be Monday, September 12th, if I'm not mistaken. And so that'll be the first exam. So you want to make sure that you do well and start the course off this semester very well. So I'm going to do an exam review right now. So these are just the questions that I think if you understand these, you're going to have a really good shot at doing well in the exam. So I posted these on Canvas. They should be up today, or Wednesday or Thursday. And you can wait to watch this if you want to work through them yourself, or you can just watch me do this. On Friday, when we do the study group, I'll either, if you have questions, I'll go through those. And if not, I'll just talk about different topics related to the exam. So let's just go ahead and get started. So these are concepts that are going to be on exam number one. So number one, what are the four major classes of biomolecules? You definitely need to know this. So one is proteins, nucleic acids, lipids, and carbohydrates. So make sure that you know all of these. So proteins, which are polymers of amino acids, nucleic acids, so DNA, RNA, and then which are polymers of nucleotides, and then nucleotides are also important in their own right. Nucleic acids. I'm going to try and have good penmanship this time. So carbohydrates and lipids. So basically all of biochemistry is going to be involving these four classes of biomolecules. So briefly describe number two, briefly describe the central dogma of biology. So the central dogma in the most general terms is that DNA goes to RNA, goes to proteins. So the information in DNA can be replicated. So DNA can be replicated, and then the information in DNA can be transcribed into an RNA transcript. And then that RNA transcript can be translated into proteins. So this is basically the secret of life. So all the information that's stored in your DNA is eventually expressed in the form of proteins with RNA as the intermediate between those. Okay, number three. So now we're talking about non-covalent interactions involved in biochemistry. So describe the four different non-covalent interactions involved in biochemistry, give a biological example of each, and then rank their relative strength. One of them is electrostatic interactions. And so this is the strongest of them. I need to remember anyway. So electrostatic interactions is one. So that is an interaction between a full positive charge and a full negative charge. So you'll have a positively charged molecule interacting with a negatively charged molecule. So a really good example of this would be salt bridges in proteins. So if say you have a positively charged lysine or a positively charged arginine or maybe even a positively charged histidine, those can form salt bridges, interactions with negatively charged glutamate and aspartate amino acids. The next one, which I think is next in strength, let me check that really quickly so I don't give you bad information. It's either hydrogen bonding or the hydrophobic effect. But so the next strongest is hydrogen bonding. And so we see that all over the place in biochemistry. So hydrogen bonding. So this is the interaction between a partial positive charge and a partial negative charge. So the partial positive charge can be something like a double bonded oxygen. That's going to have a partial negative charge. Sorry. And then that can interact with, let's say, an amino group that has a partial positive charge. And so an example of these would be the secondary structure. So alpha helices and beta sheets. So both of these are held together by hydrogen bonding interactions on the backbone of proteins of the polypeptide chain. And another good example is DNA base pairing. So in a DNA double helix, those A's interacting with T's and the C's interacting with G's, those are going to interact between the two strands on the DNA helix via hydrogen bonding interactions. And we'll talk about that in the third section, the third block of this in this class. So hydrogen bonding is the next strongest after electrostatic interactions. The second weakest or the third strongest is the hydrophobic effect. And remember that the hydrophobic effect is an entropy-driven process. So the tendency for lipid molecules or for hydrophobic molecules to interact with each other in an aqueous solution has to do with the fact that all systems tend towards disorder or an increase in entropy. So the water molecules that have to associate with two separate hydrophobic molecules, they have to have order. They are ordered around those hydrophobic molecules. Excuse me. When those hydrophobic molecules come together, that's less surface areas that the water needs to be interacting with. So those waters can be more disordered in an aqueous solution containing hydrophobic molecules. So that's an entropy-driven effect. And so some examples of these in biochemistry would be like protein folding. So in a water-soluble protein, of course, the hydrophobic amino acids, your leucines, your valines, your isoleucines, your alanines, your glycines, your methionines, and your prolines, those are, and your phenylalanines and tryptophans, those are going to be folded into the interior of the protein. And then the more hydrophilic amino acid side chains are going to be on the outside of that protein. So that is a really important concept to understand. Another important one, of course, is membrane formation. So membrane lipids have a polar head group and then a hydrophobic tail. So that polar head group is going to interact with the water molecules, and those hydrophobic tails are going to interact with each other within that membrane. And then the last interaction that we talk about and the weakest one by far is van der Waals interactions. And so these can be actually associations between nonpolar molecules. But these are when there's an induced dipole. So if you have two molecules that aren't really interacting with each other, at a certain time, the electrons may be on one side of the molecule more than another, and that can induce a dipole moment in another molecule nearby. So a really good biological interaction based on that is DNA-based stacking. So not the hydrogen bonds between the base pairs, but the DNA base pairs above and below each other in the DNA double helix, they'll actually interact via these van der Waals interactions. So that answers question number 3, the 4 different non-covalent interactions. You definitely need to know these. Definitely know that the hydrophobic effect, if you're watching this video, you took the time to study for the exam, definitely know that the hydrophobic effect is basically know everything I just told you. The hydrophobic effect is based on entropy. Salt bridges are a form of electrostatic interactions. You should probably know the relative strength and weakness of each of these types of non-covalent interactions, because these non-covalent interactions are how these biomolecules are interacting with each other within the cell and within all these systems. So it's quite important. Okay, so now we get to the math part. So I made this question really hard because it basically covers all the questions that I could possibly ask you about pH. I don't really like that section. I don't like putting math into biochemistry, but you do need to understand pH, and some of it involves math. So this question says a solution containing formic acid, which has a pKa of 3.75. You do need to know what pKa is. So it's the pH at which, I am so sorry, I keep spilling, I spilled some condensation. So a solution containing formic acid, the pKa of 3.75 has a hydroxide ion concentration of 1 micromolar. So what is the ratio of formic acid to formate in this solution? So where are we going to start with this? So we know that there's 3 equations that you're going to need to know to do well on the exam. So one is that pH equals the negative log of the proton concentration. The other one is that we know that the concentration of protons times the concentration of hydroxide ions equals 1 times 10 to the negative 14 molar squared. And then the third one is the Henderson-Hasselbalch equation. So that's pH equals the pKa plus the log of the conjugate base over the acid. Keep an eye on my penmanship. Hopefully that's legible. So what do we know? So we know the pKa of formic acid, and we know the hydroxide ion concentration. So to figure out the pH, we really need to use this equation first, because this equation is going to give us the information needed for this equation. So we need to know the pH of the solution. And to do that, we need to know the hydrogen ion concentration. Okay, so we know the hydroxide ion concentration, but you'll notice that this equation is in molar, and I gave you the equation in micromolar. So 0.1 micromolar. So let's convert micromolar to molar. So 1 micromolar is going to be 1, 2, 3. This is just the way I do it. You can do it another way. So 1.001 millimolar will equal 1 micromolar. So let's go 1, 2, 3. Boom, boom, boom. Okay, so that is 0.00001 molar equals 1 micromolar. So let's convert. The best way to do that is to convert it into a logarithmic number. So let's work backwards. So 1, 2, 3, 4, 5, 6. So the hydroxide ion concentration of this solution is 1 times 10 to the negative 6 molar. So now we can plug that into this equation right here. So H plus concentration times 1 times 10 to the negative 6 molar equals 1 times 10 to the negative 14 molar squared. So the proton concentration is going to equal 10 to the negative 14 molar squared over 10 to the negative 6 molar, which is going to equal, I'm lazy and I use my calculator in these, 10 to the 14 and to the negative 14 divided by 10 to the negative 6. Didn't let me do that. Okay, I'm having some trouble, but you can just count down. So 14, 13, 12, 11, 10, 9, 8. So the answer is the hydroxide ion concentration equals 10 to the negative 8 molar. So now we can plug that into our pH solution. So pH equals the negative log of 10 to the negative 8 molar. So that means that the pH equals, you can already guess that it's 8. Let's just put it in the calculator for fun. Yep. So pH, of course, equals 8. Okay, so now we can use the Henderson-Hasselbalch equation. Fantastic. So this is the Henderson-Hasselbalch equation. We know the pH equals 8. We know the pKa is 3.75. And then it's going to be plus log base 10 of the of the conjugate base over the acid. Okay, so subtract 3.75 from both sides. Again, I'm a lazy person. So 4.25 equals the log base 10 of the conjugate base over the acid. So let's find the anti-log. I just go like that. So 10 to the power of 4.25 equals the ratio of the conjugate base over the acid. And so 10 to the 4.25 equals a big number. So it's the ratio of the conjugate base over the acid equals 17,782, or 1.7 times 10 to the 4. And that's great. But that is actually the ratio of formate to formic acid, because this A minus equals formate. Very important to understand. And HA equals formic acid. So the ratio is actually 1 over 17,782. So 1 over 17,782 equals 5.62 times 10 to the negative 5. So this isn't too difficult. I'm not going to make a question on the exam that's actually as hard or as complicated as this one, because we don't have a lot of time. You only have an hour and 15 minutes. So I don't want you messing with, carrying the one the whole time. This isn't the point of this class. But make sure that you know how to use all three of those equations. Really important. Make sure that you know that formate or lactate or citrate is the conjugate base. And then formic acid, lactic acid, citric acid are going to be the acid form. So make sure that you don't get mixed up on that. And then also make sure that you can convert from micromolar to nanomolar to millimolar and to molar. That's really important. Why am I okay? So I'm asking you another one too. That's annoying. But I'll go through it again but we'll go through it a little quicker. So a solution contains 0.5 molar sodium formate and 0.3 molar formic acid. What is the hydroxide ion concentration of the solution in micromolar? So we're actually going to work backwards on this one. So we can start with the Henderson-Hasselbalch equation, because what we want to find out is the pH. That's how you're going to figure out the proton concentration, and then that's how you'll figure out the hydroxide ion concentration. So pH equals pKa plus log A minus over HA. We know from the last question that this has a pKa of 3.75. So pH equals 3.75 plus the log of formate, remember to keep those straight, 0.5 over 0.3. So 0.5 divided by 0.3 equals 1.66. So pH equals 3.75 plus the log of 1.66. The log of 1.66 equals 0.22. So pH equals 3.75 plus 0.22. And so that equals 3.97. So the pH of this solution would be 3.97. Okay. Already you can check that your answer makes logical sense. So the pH is higher than the pKa. That means that there's going to be more conjugate base than there is acid. If it was lower than the pKa, there'd be more acid than conjugate base. And what do we see in this question? There's 0.5 of the conjugate base and 0.3 of the acid. So there's more conjugate base than acid. So that makes sense. Check your answers at that point. Okay, so the pH equals 3.97. So now we can use this equation equals negative log of the proton concentration. So 3.97 equals the negative log of the proton concentration. Let's get rid of that negative sign. So negative 3.97 equals the log base 10 of the proton concentration. Let's do the anti-log, and I'll move up here. So 10 to the negative 3.97 equals the proton concentration. And so why did I make this like this? So 10 to the power of negative 3.97. It's about, I'm going to round this. So H plus equals about 1 times 10 to the negative 4. And so we know from our other equation, proton times hydroxide equals 10 to the negative 14 molar squared. So let's just plug in. So we know that the hydroxide ion concentration equals 10 to the negative 14 molar squared over 10 to the negative 4 molar. So 13, 12, 11, 10. So the OH concentration equals 1 times 10 to the negative 10 molar. Okay, if that doesn't make any sense to you, please let me know. But again, these will be harder than they will be on the test. Okay, this one is similar, but you don't even need to do any equations. You could use the Henderson-Hasselbalch equation, but you don't need to. So a solution of pyruvate, which is this molecule right here, it's a conjugate base, has a and pyruvic acid has a pKa of 2.5. The solution is at a pH of 7. So which of the following forms of pyruvate shown below will be present in a higher concentration? So this one you can see has the OH group. So this is HA. This is the acid. So this is pyruvic acid. So this is pyruvic acid. And then this one over here is deprotonated. And so it is a negative and it's pyruvate. So is the pH of 7, is that above or below the pKa? That's above the pKa. So the answer is that this one is going to be present in a higher concentration than the acid. So above that pKa, you're going to have more concentration of the conjugate base. Below it, you're going to have more concentration of the acid. At that pKa, the acid and the conjugate base are going to be present in the same exact concentration. Okay, so make sure that you know how to do that, especially with some amino acids like maybe histidine or something like that, because that'll be important for the exam. Okay, now we're getting to the amino acids. So you do need to memorize all the amino acids. You don't have to. This is an open book exam, so you can always just look for them, but you only have an hour and 15 minutes to do this exam. So it's better that you know the structures, the full name, the 3-letter code, and the 1-letter code of all the amino acids. It's a lot of information, but there's only 20 of them. And this will help you for the rest of the class. So in the future in this class, where I say, oh, there's a glutamate here and it's interacting with this arginine residue, you're not going to, if you don't remember them, if you aren't familiar with the different flavors of the amino acids, it's just going to slow you down and you're going to have to think about it. So if you could, you take this chance to really remember the amino acids. I promise if you do, you will be able to answer many of those questions very, very quickly. There's going to be like 4 or 5 questions on which amino acids are hydrophobic, which ones are polar but uncharged, which ones have positive charge, what's the 3-letter code of those, what's the 1-letter code of those. This will get you a lot of points very quickly if you can remember that, if you have time. So the way I like to study or practice this is just go through the alphabet. Just starting with the 1-letter code of each of these amino acids. Just start with A and work your way down to Z. And then write out the full name, the 3-letter code, the 1-letter code, and the structure of each of these amino acids. So starting with A, So is there an amino acid that starts with the letter A? Yes, indeed there is. Its three-letter code is ALA, and it's called alanine. And so what is the structure of alanine? And again, I'm going to work on my penmanship. So we have H3N plus. Then we have the alpha carbon. There's a hydrogen coming off this alpha carbon. There's a COO negative carboxyl group. And so what is the side chain? So the side chain of alanine, it's the second simplest amino acid. It's hydrophobic and aliphatic, and it just has a CH3 methyl group. So that's how I'll draw these out. I'll actually probably just make this thing and put in less detail from here on out. So the most important part is to know the side chains, of course, because the main chain is the same for all of them. Okay, moving on. That was A, alanine, three-letter code ALA. Is there any Bs? No, there's no amino acid that starts with B. What about C? Yes, there is an amino acid that starts with C. So C, CYS, that is cysteine. Okay, so we have NH3, COO, and then we have a C, and then we have an SH. So cysteine is very similar to serine, except that hydroxyl group is replaced with the thiol group. So it's a polar uncharged amino acid with an SH group after the methyl group. So that's cysteine. Okay, D. Do we have any Ds? Yes, we do have a D. So that's one of the harder ones to remember. That's ASP, and it's aspartate. Okay, so we have H3N plus, COO negative, and then we have one methyl group, and then we have a COO carboxylic acid group with a negative charge. Sorry, I had to scrunch it in there a little bit. So that's aspartate, ASP, and its one-letter code is D. So that's one of the negatively charged amino acid side chains at pH 7. So what's next? Is there an E? There is, and it's related to D. So let's move down here. So E, that's GLU. It is glutamate. So H3N plus, COO negative. And then we have one methyl group, but unlike aspartate, it has another methyl group before the carboxylic acid group, which also carries a negative charge. So aspartate and glutamate are the two only negatively charged amino acids. Aspartate comes first in the alphabet, so it has one methyl group. The glutamate comes second in the alphabet. It has two methyl groups, but they're both very similar, and they both have a negative charge. So E. Next one, F. Is there any F? There is. It doesn't start with an F. It starts with a PH, and it's phenylalanine. Okay, so H3N plus, COO negative. And so this has one methyl group, and then it has a phenyl ring, hence the name, phenylalanine. So it's basically just like alanine, except it has a benzyl ring. So this is one of the hydrophobic aromatic amino acids. There's two of them. Okay, next, EFG. Is there a G? There definitely is. So it's GLY, glycine. So the simplest of all the amino acids and the only one that's achiral. So we have H3N plus, COO negative, and then we just have a hydrogen there. So this is achiral because there's two of the same things bound to that central alpha carbon. So that's glycine. It's also hydrophobic and aliphatic. EFGH. So H is the trickier one to draw. HIS, it's histidine. So let's see how good I do this one. COO negative. So I know we have a C, and then we have N, and then there's another N. And then, so it's a 5-membered ring with 2 N's. So this has an H, And sometimes this one will have an H and there will be a positive charge. So that's histidine. So it can be positively charged at pH 7. So that's why this one's so important. So it's grouped in with the other positively charged ones, lysine and arginine, but it's not always positively charged, but it's actually really useful for enzyme activity, which we'll talk about in the next section. Okay, moving along. GHI, is there an I? There definitely is an I. So ILE, isoleucine. And so this is one of the branched chain amino acids. So we have H3N plus COO negative. And then I have to remember this one. So isoleucine is C, 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 C. I know there's 4 of them. I believe this is the structure. Yeah, so this is isoleucine. I think there actually might be one. No, there's not. So that is correct. So this is CH3. So this is isoleucine. So it's one of the branched chain amino acids. So it is definitely hydrophobic and aliphatic, and it's bulkier. So it's one of isoleucine, leucine, and valine are the more bulky hydrophobic aliphatic amino acids. Okay, moving on. HIJ. There's no J, but there is a K. So that is actually lysine, LYS. So H3N plus, COO negative. And so I believe this one has 1, 2, 3, 4, and then, oh no. I'm so sorry. I'm having some issues. Sorry, everybody. Okay, we're getting back to it. Okay, sorry about that. Okay, let's start back with lysine. So we have lysine is K, LYS, and then lysine. Let me give myself a little bit more space. So NH3 plus, COO negative. We have 1, 2, 3, 4, and then an amino group. And so this is going to carry a positive charge. So it's CH2, 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 CH2. So lysine is a positively charged amino acid. It has 4 methyl groups and then a terminal amino group that carries a positive charge. HIJKL. So L, there's definitely an L. LEU, leucine. And so this is H3N plus, COO negative. And then it's different than isoleucine, CC, and then the branch is right here. So those two are a little bit hard to keep track, but just remember leucine's got the higher branch and isoleucine has the lower branch. But again, it's one of the hydrophobic amino acids that's also aliphatic. It only has methyl groups. So HAJKLM. Is there an M? There definitely is. So that's methionine. So MET, methionine. Okay, so that is H3N plus, COO negative. And so that has 1, 2, and then a sulfur, and then a terminal amino group. Okay, so this one has a thioester group. So this group right here, but it's still considered hydrophobic and aliphatic because it doesn't have any aromatic rings and it has so many methyl groups. that it is hydrophobic as well. HAJKLMN. So N is also one. So that's asparagine. So that's one of the harder one-letter codes to remember, but I just remember asparagine. So it's N. So asparagine. Okay, and so that is H3N plus COO negative. And so that is one methyl group, and then it has a C, it has an amide group, so NH2. So that's asparagine. So it has this amide group right here, and it comes before glutamine, so it only has one methyl group, but it's actually polar and uncharged at pH 7. So LMNO, there's no O, but there is a P. So P is proline, PRO, proline. Okay, and so that's H3N plus, COO negative. And then it has three methyl groups. So 1, 2, 3. But then that third methyl group, it's the only one that forms a ring, a covalent bond with this amino group right here. So CH2, CH2, CH2. So proline's a little bit interesting. So it does break, along with serine and a few other amino acids, it breaks alpha helices pretty often. You can find it in alpha helices, but it oftentimes breaks them. And it's a hydrophobic aliphatic again, and it's the only one that has this ring structure. So that's proline. LMNOPQ, there is a Q, and so that's another tricky one to remember. So that's glutamine. I sort of put a Q sound in there, like glutamine. That helps me remember it. I don't know if it'll help you. So Q, GLN, and glutamine. And so that's related to asparagine, remember? So it's also got the amide group. So NH3, COO negative. And then we have C, but then we have another methyl group. And then we have the amide group. So just like asparagine, but with an extra methyl group. And so this is another one that's polar but uncharged at pH 7. Okay, so elemental PQR. Is there an R? There definitely is an R. So R is arginine. It's arginine. Arginine. Okay, and so that's one of the positively charged amino acids. The other one that's always positively charged at pH 7. So H3N plus, COO negative. And so that one has 1, 2, I think 3. And then it has a nitrogen, carbon, nitrogen, nitrogen. So this is also one of the bulkier ones. And it carries a positive charge on this group up here, which is called a guanidinium group or a guanidino group. So it's that carbon bonded to 3 nitrogen groups. So that's actually going to carry some resonance structure, and it'll carry a positive charge. So that is arginine R. QRS. So S is also another easy one. So, well, not easy, but a little bit simpler. It's S, serine, SER, and that stands for serine. So serine is one of those other polar one. It's very similar to cysteine. It's another one that's polar at pH 7, but not charged. So we have H3N plus, COO negative, and then we have CH2, and then that's attached to just a hydroxyl group. So of course, this is polar at pH 7, but not charged. Okay, QRST. So T is related to serine as well. So T, THR, and that's threonine. And so that is H3N plus, COO negative. And so that one has carbon. It's just like serine in that it has a carbon and an OH group, but it has one more carbon branch off of it. So very similar to serine, just with this extra methyl group right here is threonine. So QRSTU, there's no U, but there is a V. So V is valine, VAL, valine. So that's the third one of these branch chain amino acids that's hydrophobic and aliphatic. So H3 N plus, COO negative. And then it sort of just forms a V with the methyl groups. So you got C, C, C. That's how I remember it. Valine with a V sort of forms a V as well. So that's valine, hydrophobic, aliphatic. QRSTUVW. There is a W. And there's only one more after this. So let me see if I can draw this W. So W is the bulkiest letter. It's also the bulkiest amino acid. So it's tryptophan. Tryptophan. Its three-letter code is TRP, not TRY. That's the next one. So it's TRP, tryptophan. So we got, I'm giving myself some space, H3N plus, COO negative. Then we have this group. Then that's going to be joined to this indole ring. That's not a great drawing at all, but it's got this nitrogen right there. And so that forms the indole ring. That is actually a terrible drawing. But tryptophan, it's kind of the most interesting looking one. It's the bulkiest one, and it's not in Turkey any more than the other ones. So W, X, there's no X. Y, there is one, and Z, there's not. So Y is going to be our last one. So Y is TYR, and that's tyrosine, the snorkeler. So H3N plus, COO negative. So we have this methyl group here. Then we have a benzyl ring, just like phenylalanine, except it's got this OH group, snorkel sticking off the top of it, just like a snork. So that is tyrosine. So practice that for yourself. I promise you it's going to save you so much time on the exam, and it's going to make a lot of things are going to make a lot more sense later in the course if you know all your amino acids. Okay, moving on to the next question. Sorry, this is a comprehensive exam review. So describe the 4 levels of protein structure. So we have primary, secondary, tertiary, and quaternary. So primary structure. So that is going to be the specific sequence of amino acids in a protein. So that's all it is. It's just which goes where. It's a tyrosine, glycine, alanine, aspartate, glutamate, glutamine, and so on and so forth. It's just going to be the actual sequence of the amino acids. So remember that that's very important. The whole structure and function of the protein is driven by the specific order of the amino acids in the polypeptide chain. And then all the information that the protein needs to fold properly is contained within that primary sequence. So it's really important to understand that. Okay, so what is secondary? So this is the amino acid sequence. So then we have secondary structure. So this is going to be those alpha helices, the beta sheets, and the loops and turns. So all these interactions are going to be that form these secondary structures are going to be based on a hydrogen bonding between the backbone groups. So the carboxylic acid and the amino groups on the backbone of the polypeptide chain. So the side chains aren't involved. These secondary structures, these alpha helices and beta sheets, are so stable because those side chains are going to stick out. So on the alpha helices, they're sticking out of the alpha helix that's held together by those backbone hydrogen bonding interactions. And in beta sheets, those side chains are going to stick up and down, up and down, up and down, away from each other. And those beta sheets are going to be held together by those hydrogen bonding interactions. So that's secondary structure. So alpha helices and beta sheets. And it's from H bonding in the backbone. Okay, so what's the next one? Tertiary structure. So this is going to be the full folded form of that entire polypeptide chain. Just a single polypeptide chain, though. unless there's like a disulfide bond and it's multiple things, but that might be considered tertiary or quaternary structure. Anyway, tertiary structure is just the full folding of those alpha helices and beta sheets and loops and turns into a fully folded protein. And so this is going to be more driven by the side chains that are on the amino acids. So the hydrophobic side chains are going to go on the inside in a water-soluble protein. The polar hydrophilic side chains are going to be in the outside of a hydrophobic interaction. There's going to be salt bridges between these side chains. There's going to be van der Waals interactions between the side chains. There's going to be hydrogen bonding between the side chains, all sorts of stuff like that. But this is the fully folded version of the polypeptide. So fully folded polypeptide. So it's interactions with side chains. This is a very important distinction. Tertiary structure is made by interactions on the side chain. Secondary structure is interactions between the main chain. And so those are hydrophobic effect, these are van der Waals, hydrogen bonding, hydrophobic effect, and electrostatic interactions in the form of salt bridges. Those are what make the tertiary structure. So quaternary structure, what's that? So this is where multiple folded polypeptide chains can come together to make a fully functional protein. This is very, very common. It's not always the case in biochemistry, but a lot of times there's a lot of different proteins that have multiple subunits. Sometimes those will, there'll be two subunits and that protein's inactive, and then you take that subunit away and it's not active anymore, or vice versa. But the example we use is hemoglobin. Hemoglobin is only functional when it's got all four of those subunits together. So this is the interactions between multiple, so two or more folded polypeptides. Okay, so that's that question. Excuse me. So what does the Ramachandran plot tell us, and what types of secondary structure are represented? What the Ramachandran plot is, it's a way they basically looked at all the different proteins that the structures that have ever been determined, and then they looked. You remember that the peptide bond is sort of planar. It can't really can't move about that peptide bond because there's resonance structure. And that partial double bond stops it from rotating on that carbon. The alpha carbon, though, there's movement around that alpha carbon. So there can be many different ways that can move. But in reality, there's not that many orientations of that alpha carbon that are really stable when you consider all the other groups, the side groups that might interact with each other, and stuff like that. So what the Ramachandran plot is telling us is what angles of that alpha carbon are actually stable and found in fully folded proteins. And so if you sort of take these phi and psi angles, so that's one side of the polypeptide of the alpha carbon and the other side of the alpha carbon. So if you plot those in those movements in terms of degrees, you have certain regions where there's more that those angles are more common. And so one of those is associated with alpha helices. The other one's associated with beta sheets. I don't remember right off the top of my head. I think this one right here is alpha helices. And this one's like beta sheets. But you can check that. It's in your notes. It's in the lectures. It's not a big deal. So alpha helices and beta sheets are well represented in Ramachandran plots. Okay, so number 10, what kind of bonds stabilize secondary structure in proteins? So I already mentioned this. Hydrogen bonds are what stabilize secondary structure in proteins, and specifically, it's hydrogen bonds between the backbone atoms. So the amino groups and the carboxylic acid groups, not the side chain. So it's H bonding between backbone COO, which carries a partial negative charge, and then amino group. So this will be attached to something. This will be attached which carries a partial positive charge. So excuse me again. Oh my goodness. So that's what stabilizes secondary structure in proteins. What's the difference between a motif and a domain? That's a pretty good one. So a motif is like a really local sort of feature that you find in folded proteins. So these can be found in a lot of different proteins, and they're not necessarily related to the function. Sometimes they are. Sometimes there's certain like helix turn helix motifs that are pretty commonly involved in like DNA binding or something like that, but not always. So these motifs are little things that like the secondary structure will do in the fully folded protein. A domain is a part of a protein that folds independently of the rest of that protein and has a distinct function. So I talked about phosphatases, how they all, these three phosphatases all have this phosphatase domain, but then they have these other attached domains that interact with specific substrates. So that's just an example. Most proteins in your body have multiple domains that'll fold independently. So these two concepts are quite different, but you should definitely take a look at motifs and especially domains. So look at domains. So domains are part of they're both part of tertiary structure in a protein, but domains are sort of a fully functional thing. You can have individual motifs within a larger domain, if that makes any sense. What types of interactions, covalent and non-covalent, are involved in tertiary protein structure? All the different interactions. So covalent, that would be disulfide bonds. So that's a covalent interaction between two sulfur atoms on a cysteine side chain. Non-covalent, basically everything. So hydrophobic effect, because in for example, in a water soluble protein, the hydrophobic amino acids are going to fold in the inside. The hydrophilic interactions are going to be on the outside. That's really important part. Also electrostatic interactions. So if you have a positively charged lysine residue and it forms a bond, a non-covalent bond with a negatively charged glutamate residue, that would affect tertiary folding. Hydrogen bonding. So H bonding, you can also have all the polar amino acids and even the charged ones can make hydrogen bonding interactions with other side chains on the amino acids. So those are involved in the tertiary folding as well. And then also van der Waals. So just all the interactions. So a lot of times these chemicals will interact weakly through van der Waals interactions as well. So next question, what is the difference between tertiary structure and quaternary structure? Again, tertiary structure is the fully folded one single fully folded polypeptide. So that's a fully folded protein. Quaternary structure is where two or more of those fully folded proteins need to come together to make a functional protein. So hemoglobin, the pre, and then the SARS-CoV-2 spike protein were our examples in the lecture for that. Broadly speaking, how would you isolate all mitochondrial proteins and visualize these proteins? So isolating all the mitochondrial proteins, that would be through, let's be neat about this, differential centrifugation. So when you lyse open a cell, you may not, you may not lyse those mitochondria, or you can lyse it in a way where you don't lyse those mitochondria. There's also other things you can do as well. But anything that involves like a whole subcellular fraction, like we want all the cytosolic proteins, or all the nuclear proteins, or all the lysosomal proteins, or all the mitochondrial proteins, those are all going to have to, you can separate all those out using differential centrifugation. You just look up how fast you spin it, for how long, and then eventually you can get it so the mitochondria are all in the pellet at the bottom of that centrifuge, and then you just get rid of everything else. So how would you visualize those proteins? That you can just roll them on an SDS page gel. So if you have an SDS page gel, of course, this involves denaturing all the proteins and then giving them all one charge. I think it's a positive charge. And then they move through the gel in terms of their size. So their charges aren't different. The only thing different between the proteins is their size. So the smaller ones are going to move through the gel faster, and then the bigger ones are going to move through the SDS page gel slower. Remember, that's opposite from size exclusion or gel filtration chromatography. But anyway, so if you have like all the proteins in a cell, you're going to get like a smear of a bunch of different proteins on here because they're all going to separate by size. So when you run that those mitochondrial proteins, there'll be less total proteins. So it'll look something like that in the gel just because there's less of them. There's still going to be a bunch of different sizes though. So that's how you would isolate them by differential centrifugation, and then you can visualize them using SDS page. So what type of purification techniques are available, and what properties do they use to separate proteins? Let's start that. So one of them would be, let's do the size exclusion, also called gel filtration. And so that separates based on size. So you can purify larger proteins from smaller proteins by putting them through this gel. In this case, the smaller ones or the larger ones move through the gel quicker because they're not getting stuck in the gel within the column. And the smaller ones are going to come off later. So that would be separating them by size. So affinity chromatography that would be by function, I would say, or functional group. So I think the example I used is you can have a glucose binding protein attached to a bead, or you can have glucose attached to a bead, and then you're trying to purify a glucose binding protein. Well, it's going to bind to that bead, and other non-glucose binding proteins are just going to flow through the column. So that's a good way to purify your protein. A really common way of doing this is to add a 6-histidine tag. So you can take your protein and add 6 histidines at the amino acids at the end or the beginning. And then those 6 histidines will actually bind to a nickel ion. So if you have a column that's full of nickel, your protein will bind that has 6 histidines, and any other ones that don't have 6 histidines won't bind. So those are examples of affinity chromatography. And then the last one would be ion exchange chromatography. And so this would be based on charge. So you basically have a column with a material in it that's either positively charged or negatively charged. And so if your protein is positively charged, it's going to bind to those negatively charged material in the column. If it's negatively charged, you'll use a positively charged material in the column. And then the proteins with a different charge will not bind to the column, basically. So that's another way to purify your protein. You could purify it using antibodies, but that's not really a way that people usually purify proteins. That's more to detect proteins. You can isolate proteins that way, but you usually don't get a lot of them. So I'm going to move on to the next question. So these are the 3 kinds of protein purification techniques I'd want you to remember. So what types of biochemical methods use antibodies? So one would be a Western blot. And so this is, you run your protein on SDS page, but then you add an antibody. You transfer those proteins to another material, then you add an antibody and you say, oh, this protein is my protein. And I know that because the antibody bound to it. So that would be a Western blot. Another one would be immunoprecipitation. And so that is basically where you have a mixture of proteins. You add the antibody to your protein, and then you separate it out from the other proteins. So that's a way of isolating your protein. That could be considered a purification method, but it's not really used much for purification. You are purifying your protein, but not like to do other assays with, or maybe you are, I don't know. And then the last one I talked about was ELISAs. So these use antibodies to say whether a protein is present in a solution or not. And they can sort of say a bit about how much or how little of that protein is present in that solution. So those are the three main ones that I went over that talked about use antibodies. Okay, so that is it. If you have any questions, of course, let me know. I'll also be doing a study group on Friday at 8 a.m. Central Standard Time and 4 p.m. Central Standard Time, so you can ask me questions then. I can also I'll go over these questions if you want. If not, I'll just talk about different topics that will be on the exam. So I hope this was helpful. If there's anything I should be doing differently, please let me know. Happy to get any feedback. And yeah, hopefully this helps. So thank you very much.